Well, then let's talk about muscles. So looking here at our first page, got a fella, not me, of course, but somebody doing some flexing of the guns, illustrating some big skeletal muscles. But as we've talked before, skeletal muscles aren't the only muscles in our body. We have skeletal muscle attached to our bones that allows us to move and do things like take the notes that you're doing right now. But we've also got cardiac muscle inside our heart and smooth muscle found in our organs like the stomach, the intestines, and the bladder, the uterus, and so on and so forth. Those different forms of muscle contribute to homeostasis either through sustained contraction or by alternating between contraction and relaxation, providing us with two key functions from our muscle tissue. As far as the first two functions there, producing body movement, stabilizing body positions, that relates specifically just to skeletal muscle, okay? You can see examples of those are listed there, probably pretty straightforward. Number 3 there, moving substances within the body, that relates to the other two types of muscle. So obviously heart or cardiac muscle pumps blood, and then the other bullets there relate to smooth muscle, moving substances in the digestive tract. We have sphincters preventing outflow of contents from those hollow organs, movement of sperm and oocytes or egg cells through the reproductive system, urine, the urinary system, through the lymphatic systems. And then the generation of heat, it really truly relates to all three types. But when you think about how much muscle there is when it comes to smooth and cardiac, it's pretty minuscule compared to the amount of skeletal muscle. So the amount of heat that comes from contracting muscles predominantly coming from skeletal muscle. And we've talked about this before, but we shiver when we get cold, and that's because when we contract our muscles, we produce heat. That process is, or that term is called thermogenesis, and literally translates to mean heat formation or heat creation. And that's why we shiver, because when our muscles contract, and what we're only talking a lot about in this lecture, how muscles contract and all the different processes that go into that, a significant amount of heat is produced. Although bones provide leverage and form the framework of our body, they cannot move body parts by themselves. Motion, therefore, results from alternating contraction and relaxation of muscle, which makes up about 40 to 52% of total adult body weight. And like I said, by far and away, the most type of muscle that we have, the major type of muscle that we have, is skeletal. So that 40 to 50% really truly relates to skeletal muscle most. Okay. Your muscular strength reflects the primary function of muscle, and this can be said for all three types, transforming chemical energy into mechanical energy to generate force, perform work, and produce movement. And yes, we'll be talking about ATP and its role in allowing for us to perform work and generate force and produce movement, because that's what the chemical energy that our muscles use. ATP, as we know, we've talked a lot about before. We'll see again here today. Okay, really quickly, I just wanted to kind of go through each of the three types of muscle. For those of you who are in anatomy, this is stuff we did in that class a few weeks ago. Most of what we'll be talking about in this lecture specifically relates to skeletal muscle, but I wanted to kind of talk about each of the three types just briefly. We'll get into obviously more about heart muscle, cardiac muscle in our next lecture when we get to the heart. And then as we roll through lectures in the future, where we talk about like the bladder and the uterus and the stomach, we'll talk about the smooth muscle there. So really quickly, let's just talk about the three types. Skeletal muscle is named because it is attached to our skeleton and thus moves our bones. Key feature of skeletal muscle, and we'll see the same feature in cardiac muscle, is that it's striated. And this is why this picture looks like it is kind of fuzzy. You can see these light, dark kind of fuzzy lines. So that's not an accident. That's actually what it looks like under a microscope. Those light, dark bands are called striations, and they are only visible when looking at skeletal, and the same thing for cardiac muscle when zoomed in under a microscope. Okay. Key feature of skeletal muscle is that it works mainly in a voluntary manner, and that is because it's regulated by, as we know, the somatic nervous system, and that means its activity can be consciously controlled, although most skeletal muscles are also controlled subconsciously to some extent. meaning we don't have to like dedicate all of our brain mental capacity towards doing a movement. For example, like the diaphragm, that muscle that allows us to breathe, we really never think about that. And it's contracting, relaxing without our really conscious awareness. And the only time we ever think about it is like if we want to take a deep breath, or if we actually want to like hold our breath, like if we go underwater. Skeletal muscle obviously attaches to bone, but also in some places it attaches to skin, connective tissue called fascia, and even in some cases other muscles. We're going to describe skeletal muscle as being multinucleated, which means that there are more than one nucleus per cell. In fact, you can see like right here where the number 1 is, right here where the number 1 is. That's a nucleus. There's a nucleus, another nucleus. You can see lots of nuclei in each individual cell. And the nucleus is, or nuclei are always going to be pressed up against the plasma membrane. Cardiac muscle found only in the walls of our heart in a layer known as the myocardium. Like I said, we'll learn more about this tissue in our next lecture, which is a nice transition from skeletal muscle into cardiac muscle when we talk about the heart. Like I said on the previous page, it's striated like skeletal muscle. A key feature that cardiac muscle tissue has that the other two do not. You can see these dark bands right here. Those are called intercalated discs, and they are basically just a thickened plasma membrane between the two cells that hold some special features that we'll find out more about later on in this unit. They're uninucleated, so just like most cells of the body, there's only one nucleus, and the nucleus, instead of being pressed up on the outside of the cell, is more embedded on the inside or within the middle of the muscle fiber. Action of cardiac muscle tissue is strictly involuntary, and the innervation for that muscle contraction that takes place and the relaxation that takes place actually originates from within the heart itself in a node of tissue called the pacemaker. The pacemaker is a structure called the sinoatrial node that we'll learn more about. You've probably heard of a person having an artificial pacemaker put in. That helps regulate the heart rhythm in some way that their cardiac conduction system is not correctly working, and they've got a regular rhythm or a regular beat to their heart. This built-in ability of the heart to initiate its own contraction is termed autorhythmicity, and that's because auto means self and rhythm, rhythmicity for rhythm. If the heart was left alone, it would keep a resting heart rate at 100 beats per minute that would never change. Obviously, that 100 beats per minute is not what our heart rate is always at. It can go up, it can go down. In fact, hopefully any person is not going to have a resting heart rate nearly that high. And that's because the pacemaker is where the signal originates from, but the pacemaker can be influenced from outside sources, things like hormones and neurotransmitters. Like for example, we've talked about epinephrine, which is, as we commonly know it, adrenaline. What's that going to do to your heart rate? It's going to make it go up. Okay. And same thing for other chemicals like acetylcholine. That's going to make it go down. So the signal comes from the SA node, but that signal can be altered as far as how often it occurs. And we'll learn more about that later in this unit. And as we've previously learned, cardiac muscle tissue can be regulated by the autonomic nervous system, like the sympathetic parasympathetic stuff that we hit hard a few weeks ago. Smooth muscle tissue, which we won't talk much about in this unit, is located in the walls of hollow internal structures like your blood vessels, your airways, and many organs, the stomach, the intestines, the bladder, the uterus, etc. It's called smooth muscle because it's going to lack the striations that you find in skeletal and cardiac muscle tissue. The nucleus is located within the muscle cells like it is in cardiac, and the same thing, it is uninucleated, so one cell, one nucleus. Usually is involuntary. A key feature of smooth muscle is that's relatively slow moving, but really resistant to fatigue. So it doesn't get tired nearly as easily as other muscle tissue, especially skeletal muscle tissue, which is fantastic. Like, if your stomach is working hard to digest, a big lunch or a big dinner that you had, it's not going to get tired and worn down. And just like we previously learned about with cardiac muscle tissue, smooth muscle tissue is regulated by the autonomic nervous system. Now, regardless of which type of muscle you're talking about, whether it's cardiac, whether it's skeletal, or whether it's smooth, they all have 4 very important special properties that enable them to maintain and promote homeostasis. And those 4 properties are listed here that you're filling in now. Some of these properties might ring a bell to you. Hopefully that first one does. We talked at great length about excitability previously. Excitability, once again, we've seen this before, but it's the ability to respond to stimuli by producing electrical signals known as action potentials. Excitability is a property that not only muscle tissue cells have, but also neurons. Okay. In muscle tissue, 2 types of stimuli can signal an action potential. And I've already talked about one, autorhythmic signals, which arise in the muscle tissue itself, in that case, like the pacemaker of the heart, or in what we'll see in this lecture when we talk about skeletal muscle, it's chemical stimuli, such as a neurotransmitter released by neurons. We'll talk a lot about a familiar neurotransmitter we've talked about quite a bit previously, acetylcholine. Could even be hormones distributed by the blood, or even just a local change in pH. Contractility, this is a unique property to muscle, and the word contract really is the beginning of that word, and that's essentially what contractility means, the ability to contract forcefully when stimulated. So in other words, when a muscle tissue cell, once again, whether it's cardiac muscle, skeletal muscle, or smooth muscle, doesn't really matter, when muscle cells get excited, they contract. Okay, so when a nerve signal or action potential is created in a muscle cell, it's going to respond by contracting. And when a muscle contracts, it generates tension while pulling on its attachment points. If this tension is enough to overcome the resistance of the object we move, muscle will shorten and movement occurs. And that's really all just physics. It has to be with regards to movement, effort greater than resistance, and stuff I'm not really going to get into. Extensibility and elasticity are properties that, for those of you who in anatomy or taking anatomy, have learned that, for example, your skin exhibits. Think of muscle tissue as far as extensibility, elasticity go, like a rubber band. They have the ability to stretch. That's what extensibility is. Think extents or extend. It gets longer. Okay. Now it says there without being damaged, obviously, just like a rubber band, if you stretch something too far, it's going to snap. Okay. And once again, we're not just talking about like stretching your muscles in your joints and your legs, like when you do before a run. We're also talking about like your cardiac muscle that has to stretch when the heart fills up with blood, or the stomach, the smooth muscle in there has to stretch when it fills with food. And just like a rubber band, after being stretched, or if you take a rubber band and you crinkle it up and you let go, it returns to its original length. That's what elasticity is, the ability of muscle tissue to return to original length and shape after contraction or extension. And elasticity also goes by the term elastic recoil. Those 2 properties are not only important in muscle and, as I mentioned, skin, but one of the other most important places we have extensibility, elasticity, as we'll find out later in the semester, is our lungs. So we'll get into that at that point, but that's a few weeks away. Now, what we're going to do here for the next couple slides is talk about some anatomy of skeletal muscle. And like I said, I just wanted to kind of introduce the 3 types of muscle, but the pretty much remainder of this lecture is all about skeletal muscle and how it works, as we'll move on into the other types later on. To begin talking about skeletal muscle, I wanted to kind of introduce the anatomical features because, you know, we do this a lot in this class. It's important to understand structure because it affects function. Okay? Although this is not anatomy, it's important to have a basic understanding of how things are built, in this case, skeletal muscle. Most important components of a skeletal muscle are the muscle fibers themselves. The term muscle fiber is just another way to describe muscle cells. Okay, so what we're going to find is we're going to see some new terms which are technically not necessarily new because they just have a different meaning from a term you learned earlier in the semester. Okay. The diameter of a mature skeletal muscle fiber can range from as small as 10 to as large as 100 microns. Okay. Now, what's really cool is the number of skeletal muscle fibers that we have is actually set before we're born. Okay. So in other words, for a muscle to grow in size, muscle fibers don't grow in number. They don't go through cell division like what our bone cells do to increase your bone size. Instead, the muscle fibers increase their size, a process called hypertrophy. And the opposite of hypertrophy is atrophy, which is where muscle fibers will shrink. Okay, and that's common, like if muscles aren't getting used. You know, if you've ever spent time in a cast and you take that cast off, you know, you might see that arm or that leg really shrunk up. That's not because the muscles have disappeared. It's just the muscles have kind of gotten smaller because the muscle fibers have gone through atrophy. And thankfully, hypertrophy can be quickly enacted to allow those muscles to get back to their original size. Hypertrophy can be stimulated by hormones we previously learned about, growth hormone and testosterone, which explains why you see a big increase in growth of muscles in boys and girls when they hit puberty, especially boys, because not only are they producing lots of growth hormone, that's really when men start producing testosterone. And I guess I shouldn't say men, but I guess they are. If they're going through puberty, they're becoming a man. So that is why men typically have bigger muscles than women. I say typically, not always. Now, as skeletal muscle fibers mature, they originate as embryonic stem cells known as myoblasts that then fuse together, forming a multinucleated muscle fiber. And we can see an illustration of that here on the next page. I'm going to let you fill in the blank here on this page before I kind of talk about the figure. So up here in the top left of our illustration, we see these individual cells called myoblasts. These are the cells that eventually will form our muscle fibers. What happens during, and this is all embryonic, so before the baby's even been born, those myoblasts fuse together. And what you're left with is a singular cell where all the nuclei of those original cells are still retained. Okay. Now, after we're born, and we have our muscle fiber number set, we still can change the makeup of these muscle fibers and the amount of nuclei we have thanks to what we call satellite cells. Okay, satellite cells are just like supporting cells that help increase the number of muscle fiber or of muscle nuclei. Okay, now where we find those nuclei, I've mentioned earlier back on this slide, is that typically they are pressed up against the cell membrane. Okay. Well, we're going to use a different term, what we call the sarcolemma, to describe this plasma or cell membrane of a muscle cell, which if we look at this bottom image is shaded blue. Okay. This is just a fancier term for something we previously learned about earlier this semester. That lipid bilayer made up of phospholipids, and you've also got glycolipids and proteins and whatnot in there. Now, we just got done talking about neurons and the nervous system not too long ago. The plasma membrane or sarcolemma of a muscle fiber is very similar to the plasma membrane of a neuron in that it maintains a resting membrane potential. And the resting membrane potential was that -70 number that our neurons have. And if altered, that resting membrane potential has a threshold which can trigger the formation of an action potential. Well, muscle fibers also can do the exact same thing. Okay? They can alter their resting membrane potential that then will trigger an action potential. And just like in a neuron, action potentials can spread from their original site. Remember how like in a neuron, it goes down the length of the axon? Well, in the case of an action potential in a muscle fiber, that action potential is going to spread throughout all of the sarcolemma on the outside of the cell. But more importantly, what we need to do is channel that action potential to the center of the muscle fiber, which is thankfully going to occur because of these little blue tube-like structures, which are called T-tubes. T-tubes is what I usually call them. That's short for transverse tubules. Their job is just to take an action potential from the outside of the cell to the inside of the cell. Okay. Now, the T-tubes are colored blue here, but you see on both sides of T-tubes and kind of all throughout the interior of this muscle fiber is a gold shaded structure, what's called the sarcoplasmic reticulum. Okay. The sarcoplasmic reticulum is a fluid-filled system of membranous sacs that encircle what we call myofibrils. I'll talk about those here in a second. Okay. The sarcoplasmic reticulum sounds a lot like the endoplasmic reticulum that we learned about earlier in the semester. Well, that's because it's actually just another way to describe the SER, smooth endoplasmic reticulum, that we find in non-muscular cells. Okay. Now, when we learned about smooth endoplasmic reticulum, that organelle, when we talked in our cells lecture way at the beginning of the semester, we learned that the smooth ER does a lot of different things. Okay. In our muscle cells, the most important thing it does is it stores calcium ions. Okay. The main function of it is not just to store that calcium ion or those calcium ions, but to then release them when a muscle needs to contract. Okay. When that action potential spreads into the center, thanks to those T-tubes, the sarcoplasmic reticulum dumps its calcium out into the cytoplasm of the muscle fiber. And as we'll find out in a little bit, calcium is absolutely essential for a muscle to contract. Okay. If you look really hard, you can see that on both sides of a T-tube are fattened ends of the SR. That's what these structures labeled here are called terminal cisterns or terminal cisterna. Okay. That's where you're going to find calcium channels that literally are called calcium release channels that release calcium that then helps trigger a muscle to contract. Okay. Within the sarcolemma is the sarcoplasm. This is just a fancier term for the cytoplasm that you find in other cells. Okay. It contains a substantial amount of stored glycogen. Remember, glycogen is just bonded glucose molecules together that we can then break down and release to make ATP with. That glycogen is stored in vesicles known as glycosomes. Those aren't visible in that picture, but that's okay. There's also a red colored protein called myoglobin in the sarcoplasm. Okay, myoglobin is a lot like another protein you may have heard of before. Gets a lot more publicity, and that protein is called hemoglobin, which is found in our red blood cells, and actually is the reason that our red blood cells appear red, and that's why our blood appears red. Okay, I always tell students to think of myoglobin and hemoglobin like cousins. Okay, they both are a red colored protein, but more importantly, they have an important role with oxygen. In the case of hemoglobin, that's the protein that helps us transport oxygen. In the case of myoglobin, it can bind oxygen molecules so that oxygen can be released to make ATP. Okay. In fact, it is what actually is going to cause like steaks and like hamburger to appear red, and then when you cook it, if you don't cook it all the way through, as they say, you're going to have a steak that's bloody. It's not actually blood what you're seeing, it's just a red colored fluid because the myoglobin hasn't broken down. And so if you ever hear somebody say that their steak is bloody or they like their steak bloody, you can tell them they don't know what they're talking about. Okay, now myofibrils are these red tube-like structures that you can see here in our figure. And we can describe these structures as basically the contractile organelles of a muscle fiber. Okay, they are thread-like structures, so they're thread or rod-like. In fact, here you can kind of see why they call them thread-like. This is a figure of a muscle fiber and opened up and zoomed in, what, 720 times what the naked eye can see. This is an actual real one. You can see these little kind of stringy structures. Those are the myofibrils, and those are what do the contracting of the muscle fiber, which therefore contracts and shortens the entire muscle. Okay. As you can see here in this figure, and you can see down here in this figure, Myofibrils run parallel to one another, and they actually extend the entire length of the cell. Okay, it's the reason that these guys are built the way that they are that result in skeletal muscle fiber, and we'll see the same thing in cardiac muscle fiber. That's why they're striated. Okay? And it has everything to do with these structures called sarcomeres and the thick and thin filaments that you can see that are labeled here. And we'll talk about those here on this page. Okay, good. So within muscle fibers or within myofibrils are smaller structures called filaments. Okay, and there's two major filaments, what we call the thin filaments and the thick filaments. Okay, and they're named because one's thin, one's thick. It's as simple as that. They're both the same length, about 1 to 2 microns. Okay, the thin filament's about 8 nanometers in diameter, which is about half as thick as the 16 nanometer diameter thick filament. Okay, those are the structures that are directly involved in the contractile process. Okay, well, if I go back to this previous page, we said that myofibrils are parallel to one another and extend the entire length of the cell. Okay, but thick and thin filaments do not run the entire length of the myofibril. Instead, they are organized into compartments known as sarcomeres. Okay, and a sarcomere can be described as the basic functional unit of a myofibril. Okay, if we look at this next page as an illustration of, at the top here, we can see kind of a 3D rendering of at least part of a myofibril. And we can see bracketed off here is a singular sarcomere. Okay, a sarcomere is just where you find the thick and thin filaments. Okay, and what you'd find is that one myofibril is going to be made up of lots and lots and lots of sarcomeres. They're just right next to one another. So here you'd have a sarcomere, then you'd have one here, and then you'd have one here. If this figure kept going in both directions, you'd see sarcomeres all over the place. Okay. Well, if you look inside of a thick and thin, or excuse me, inside of a sarcomere, what you're going to find is that the thick and thin filaments are in specific places where you will find in some areas only the thin filament. In some areas, you'll find only the thick filament. And in some areas, like right here, they are overlapping one another. Okay. Now, obviously, this is dependent upon whether the muscle's in a contracted, a relaxed, or a stretched state. Okay. But this creates these different zones and bands. And this is where the striations come from. Okay. Let's first talk about the Z discs. Okay, Z discs, which are nice and zigzagged here in both of our figures on this next page, which is how I always tell students to remember, Z discs are zigzag. Okay, Z discs create the borders of a sarcomere. So in other words, a sarcomere goes from one Z disc across to the other Z disc. And then you'd find another Z disc over here, and that would be its sarcomere. Okay, what is a Z disc? Well, aside from forming the borders of the sarcomere, it's also very important because it's made up of a dense protein material, serving as attachment point for the thin filaments. Notice here, all three of these thin filaments here, all three of these thin filaments here, these two, these little thin red lines are all connected directly to the Z discs in our image. Okay. Now, on both sides of a Z disc, you're going to find a segment of the sarcomere where there's only that thin filament. We call that segment the I band. Okay, this segment includes Z discs and the ends of the thin filaments where they do not overlap with the thick filaments. Okay, whereas the I band is only thin filaments, the A band is where you find the thick filaments. Okay, and the A band is typically the dark middle part of a sarcomere. Okay, but the A band actually has 3 zones within it. Okay, 2 what we call zones of overlap and 1 H zone. Okay, the H zone is strictly thick filament only, whereas the zones of overlap, exactly what it sounds like, are the segments within a sarcomere where the thick and thin filaments overlap one another. Okay. Once again, it's this arrangement of thick and thin filaments within a sarcomere that cause a muscle fiber to look striated when under a microscope. In fact, the I band is what creates the light striations. The A band is what creates the dark striations, which is kind of handy. I is in the letter, or is in the word light. A is in the word dark, so it goes light, dark, light, dark, light, dark. And if you go back to this illustration or picture, I should say, you can see those light, dark bands over and over and over again. Okay. Last part of the sarcomere, I guess I haven't talked about, is what we call the M line, and that's basically pretty common sense as to probably why they call it the M line. It is the middle, it is the direct center of the sarcomere, and this is where supporting proteins hold the thick filaments together at the center of the H zone. Now, in anatomy, you guys had to learn and identify all the different parts to a sarcomere, so this is a review for you guys. But, you know, in this class, I'm not so worried about you identifying those. Just having an understanding of it is important, okay? Now, there is a third filament that you can see listed here called titin. We'll talk about titin here in a little bit. It's a type of protein, and it serves a very important function. One thing I want to keep in mind before I move on is, you know, this illustration is fantastic. It shows all those zones and the structures very nicely. But I really like what they've done up here and what they've shown here to demonstrate that these things are three-dimensional structures. And we're not dealing with a piece of paper that's, you know, doesn't have any depth to it, where there's only 2 dimensions. We've got 3 dimensions. So there's depth and thickness to it. All these big red dots, that's a thick filament, okay? All these little smaller red dots, those are thin filaments, okay? So you have to keep in mind that we're not just dealing with something 2-dimensional, it's something definitely 3-dimensional. Now, within a myofibril, you're going to find 3 types of proteins, okay? The first type of proteins we'll find are contractile proteins, okay? And these are the 2 major proteins we'll spend the most time talking about, what we call myosin and actin. Okay? Then you've got regulatory proteins, and the third type of proteins are called structural proteins. Once you guys are done writing things down, I'm actually going to go ahead to the next slide where I've got an illustration, or several illustrations actually. So I'm going to kind of bounce back and forth between these next couple slides. Okay. All right, here on this next slide, we can see at the top left one of the 3 contractile proteins, the first, or one of the 2 contractile proteins, and that's myosin. Okay, myosin, along with actin, are the 2 contractile proteins, which once again we'll spend the most time talking about, called contractile proteins because they are directly going to generate the force during contractions. They're the ones that are involved with the muscle contraction process. Okay, myosin is a motor protein found in all 3 types of muscle tissue, so it's not just in skeletal, but it's also in cardiac and smooth. Okay, these are proteins that will pull or push various cellular structures to achieve movement by converting chemical energy in the form of ATP into mechanical energy of motion. Okay, one singular myosin molecule looks like this, where it's kind of like taking 2 golf clubs, twisting the shaft around one another, so it's very tightly joined to one another, okay? And then their heads are found at the end, like a golf club. But very different than a golf club, the heads of a myosin molecule have a hinge, okay, which enables them to do like what your wrist can do or what your elbow can do. It can extend itself and it can flex itself, or it can bend and straighten, okay? So like right here, we can see these two heads are not in the exact same position. One's a little bit more extended, one's a little bit more flexed. That's because of the hinge that they have. Now, why do they hinge, or why do they flex and straighten out? Well, energy alters their shape, okay? In fact, on both of these heads is a site where ATP, that energy, can bind to it to energize it, okay? When ATP binds to a myosin head, it's going to enable it to eventually grab and pull on an actin, okay? In fact, literally at the very tip of the heads will be a actin binding site, which name tells you exactly what its job is to do, attached to a myosin. In fact, myosin, or excuse me, attached to an actin, actin molecule, each of these little beads has a myosin binding site, and that's where they can come together and form what's known as a cross bridge, okay? Now, here's what one of the thick filaments that you're going to find in a sarcomere will look like. Remember, there's not going to be just one. You can see here in our illustrations there are multiple, or if I go back to this image, we see a lot of them three-dimensionally arranged, okay? One thick filament is made up of about 300 of those myosin molecules, okay? So all of these little kind of things sticking out, those are myosin heads. And so what you've got is a whole bunch of these guys forming one thick filament, okay? Some of them are arranged going in this direction. Some of them are arranged going in this direction. Right here in the middle, that would be the M line made up of the protein that we just talked about that kind of holds them in place, okay? Now, what's wonderful is that these heads, it's kind of hard to show, but they point about 45 degrees away from one another, maybe even like 60 degrees, okay? And what this is, why this is important, because once again, you have to think we're dealing with something three-dimensional. You've got all these different heads pointing in different directions, which is very nice because as you can see back on this illustration, see all these little tiny red dots? I said those are thin filaments. Okay, each of those thin filaments, there's 6 of them around each thick filament. Each thick filament has those heads pointing it off in different directions, and thus can grab and pull on all those different thin filaments. Okay. When it comes to the thick filament, myosin is the only protein making it up. That's not the case for the thin filament. Actin is the main component of the thin filaments, but tropomyosin and troponin are also a component, okay? Well, let's first talk about actin. There's 2 forms of actin actually, and it's really not that important to understand, but I wanted to kind of explain it because it's shown in the illustration. We have what's called G-actin and F-actin, okay? G is short for globular, F is short for filamentous. Basically, it describes kind of like if you were to take beads and make a necklace, okay? Each bead is what like a G-actin is, okay? F-actin is like taking the beads, putting it on a necklace, and there's your necklace. That's the F-actin. Instead of being one-stranded necklaces, they're a helix arrangement where there's 2 strands, and they're in a helix. They're coiled like this, like DNA was that we learned about earlier in the semester. And as I mentioned earlier, each of those beads, each of those G-actins has a spot where myosin can grab onto it, okay? Now, what takes place is a myosin head will attach, forming what's called a cross bridge. It's going to want to attach itself when it's in this position though, okay? Because then it can grab and pull itself. So it goes like when you flex your elbows or flex your wrists, it's going to grab and pull, and then it'll go from this position to this position. And that's what pulls on the thin filament, okay? If you have like something tied to a rope and it's all the way across the room and you want to pull that structure to you, you're going to take your hand and you're going to grab the rope with your arm extended. And then you grab and you pull by bending your elbow, and that causes the rope and the structure that's attached to whatever it is to come closer to you. Okay, and then you want to grab with your other arm and pull, and grab with your other arm and pull, and grab with your other arm and pull. That's what these two heads end up doing. What they do is they constantly grab and pull and then break, grab and pull and break, grab and pull and break, grab and pull and break. And that's going to be what causes the sarcomere to shorten. Okay, over here on the right side of the page, we show what happens to sarcomeres when you go from relaxed to partially to maximally contracted states. Okay, if we look here at the top, we've got the H zone where there's only the thick filament highlighted green, the I band where there's only blue, or where there's only thin filament highlighted blue, and then the A band highlighted pink where there's the entirety of the thick filament. Okay, looking here from relaxed to partially contracted, what's taken place to the H zone and the I band is they've gotten smaller. Okay, what takes place is the sarcomere gets shorter because the Z discs end up getting closer to one another, because those thick filaments are grabbing and pulling the thin filaments towards the M line. Okay, and so the area where there's only thick filament, the area where there's only thin filament, those start to get smaller. Okay, because eventually what happens is the zone of overlap is growing. What we see not occurring is any change to the A band. In other words, the length of the thick filament does not change, nor does the length of the thin filaments. The filaments don't get shorter, the amount of overlap is what alters. Okay, and that's the reason why we call this process, where's that? I missed it, I think. There it is. The sliding filament theory that states actin and myosin filaments don't get shorter during contraction. Instead, they slide over one another, and that's what causes the muscle fiber to shorten. We'll get into all that on Thursday. I'm jumping ahead, but that's okay. So that's myosin and actin, the two most responsible for muscles to contract. But as I've already mentioned, actin is not the only component of the thin filament. In fact, down here at the bottom, we show that the other two proteins, tropomyosin and troponin, are going to play a big role in muscle contraction as well. Tropomyosin and troponin are what we call regulatory proteins, and they turn the contraction process on or off. Okay, and this illustration down here at the bottom, this would be a situation where the muscle would not be able to contract. Okay. And I can tell you that because those thick are on the actin, all those myosin binding sites, they're covered up by a protein called tropomyosin. Tropomyosin is kind of like a rope covering those binding sites. When calcium, that we learned earlier, is released from the sarcoplasmic reticulum, it binds to the troponin, the other regulatory protein. And when it does, what's going to happen is the tropomyosin will get out of the way. It'll shift over and expose those myosin binding sites. And that's when the myosin heads can come and attract or attach, forming what's called a cross bridge. We're going to get into all that on Thursday. The third and final type of protein that you find in a myofibril is what we call structural protein. Okay, their job is to kind of keep structure, hence the name. They keep the thick and thin filaments in proper alignment, give the myofibril its elasticity, extensibility, and can link the myofibrils to the sarcolemma extracellular matrix. If I go back to this illustration, I mentioned titin was a filament that I didn't talk about, but I pointed it out. Titin is the third most plentiful protein in skeletal muscle, coming in after actin and myosin. Gets its name because of its large size. That's literally what titin means, really big. Its job is to stabilize the position of myosin by spinning from a Z disc all the way to the M line. So it runs from the Z disc to the M line and just helps hold the position of the thick filament. Okay. It is very important, aside from doing that, because it accounts for much of the elasticity and extensibility of our myofibrils. In other words, it accounts for the ability to stretch and recoil. Okay. Stretch for extensibility, recoil elasticity. Dystrophin is going to link thin filaments of the sarcomere to integral membrane proteins in the sarcolemma. You may have heard of the condition called muscular dystrophy before. Unfortunately, a congenital, typically anyway, condition, disease that affects pretty young kids. The most common type is called Duchenne's muscular dystrophy. It results from an inability to produce the correct type of dystrophin. It's essentially a faulty dystrophin that's produced, and thus over time the muscle starts to become weak and broken down, and eventually the person passes away because their muscles stop working, like the ones that enable them to breathe. So unfortunately, it's a really, really bad deal. Nebulin, final type there, wraps around the entire length of each thin filament, helping to anchor the thin filaments to the Z discs. Okay, now that was a quick rundown on the anatomy and kind of the structural makeup of skeletal muscle fibers, even at the most smallest microscopic level. But it's important to have a good understanding of structure, because on Thursday we're going to start by getting into the fun stuff when muscles contract. So we'll stop here for today. This is a perfect spot to digest what you just learned and dive in with some fun stuff in a couple days.